Hi, in this video, I'm just going to discuss the DNA replication videos, both the simplified version and the realistic one, just to iron out the nuances of each of the videos. So here we start with the simplified uh, video. So you have here a model of the DNA, and the model is again simplified, so you don't get to see the individual structures of the sugar phosphate backbone, for example, and the nitrogenous bases. Rather, they are just represented by shapes. So here they're color coded. So you have the backbone, sugar phosphate backbone, up and bottom, and then you have the bases, guanine, cytosine, adenine, and thymine. Now, if you remember, the DNA is double-stranded, and again, it's anti-parallel, meaning one strand goes in the opposite direction as the other strand. So in this case, we can see that this upper strand is the 5' to 3' end if you go from left to right, and then the bottom strand is 3' to 5' again if you go from left to right. So in DNA replication, what happens at the start is, again, you have the enzyme helicase, and the helicase kind of splits the DNA into two. And again, just to emphasize this is one of the unique things about DNA replication. So the DNA replication, when you have the DNA, which is like, again, a ladder, right? Instead of, this is the old DNA, and then you create a new DNA from scratch, such that this now is the new one. You don't do that. What you do is you have the old DNA, which is this one, and then what you do is you split the DNA into two, right? You split it into two, and then now you build the other half to complete a new DNA strand. So again, that's unique. It's not like when you photocopy. Again, I used photocopying as an analogy, but photocopying is you're producing something new from something old, and you're using the old as a template. But this is really old, and that's new. This, in the DNA replication, you're splitting the DNA into two, and therefore the old strand remains, and then the other half is what the one you're building. So that's why we call this process a semi-conservative process, because technically speaking, you have two new DNA strands, but I mean two new DNA molecules, but in that DNA strand, in the DNA molecules, one strand of it is old, while the other strand is new. Okay, so this is again, I want you to have kind of a narrative to your story. So when you, for example, have to remember that in the first process, the first step of the process, the helicase, it will split the DNA into two. Why would it split into two? Because again, that's how DNA is replicated. It's not like you're making new DNA molecule from scratch. You are using half of the DNA of the old DNA as a template. Okay, so you have there a replication fork. So these terms you have to remember. You have to remember helicase and then replication fork. And then here, this strand, um, you have the primase that attaches to it. So think of it as the primase job is to determine where to start in the replication. And the way it's done is that primase molecule attaches RNA bases. So this is RNA. So if you notice, um, there are subtle differences here in the animation. See, it's not light blue. The backbone is not light blue. So the backbone is kind of darker blue. So this means that this is a sugar phosphate backbone. This is a sugar phosphate backbone, but the sugar phosphate backbone here is deoxyribonucleic. So deoxyribo sugar and then phosphate. But here, the backbone is ribose and phosphate. So ribonucleic. So this is RNA. And if you can notice, instead of red, you have brown. So this red here represents thymine. But in the primers, we call this the primer. In the primer, there's no red. Instead, it has kind of brownish orange or brown, and that is representing uracil. Okay, so what's the narrative? The narrative is you have to know where to start, and the primase kind of tells the whole process where to start by attaching what we call primers. Those are primers, and then once the primers are in there, the DNA polymerase, which is the next enzyme. So right now we have three enzymes. First was the helicase. Second is the primase. This one is the polymerase, and the polymerase now knows where to start, right? And then what it does is the polymerase now then starts to build the second half of the DNA bases by again base pairing it A to C, C to G, but more importantly, this direction is 3 prime to 5 prime. So notice that the DNA polymerase always starts from the 5 prime end moving towards the 3 prime end. Okay? And that is later on important in our narrative. So understand that a DNA polymerase is kind of limited in the sense that it can only do 5 prime to 3 prime direction, but it cannot do 3 prime to 5 prime direction. Okay? More in this later why it cannot do, or an analogy why it cannot do 3 prime to 5 prime. But for now, just remember that it will only build from 5 prime to 3 prime. Okay? So before we go further, let me point out some things here. Like, okay, sir, the DNA polymerase builds the bases, but I noticed that the bases are kind of being sucked in from somewhere. So the bases are just floating, from the material in the nuclear in the nucleus, because presumably these chromosomes are in the nucleus, and they're just floating, and it seems like the DNA polymerase is just sucking them out from nowhere. And again, this is where we will connect this idea to your digestive system. So you see, when you eat food, food is essentially plant material or animal material. Like you eat chicken, you eat pork chop, that used to be pig, that used to be chicken, that used to be rice, which is a plant. My point, those things are living things. So if these are living things, guess what? They have cells. Their cells have nuclei, and inside those nuclei, you have DNA. So, meaning when you digest the plant material and animal material, you are literally dismantling all of those chemicals that are inside the cells, including the DNA. So, when you're eating, the DNA material from the plant or animal that you ate will actually be shredded into nucleotides. And these nucleotides will just be distributed by your digestive system via your circulatory system to all of the cells in your body. And therefore, these nucleotides will literally be in the cell material, literally just floating in the material. It's probably in the cytoplasm, or maybe probably in the, most definitely in the nuclear materials, right? Inside the nucleus, because this process happens inside the nucleus, right? And probably also in the cytoplasm. So the idea is these nucleotides are just floating around, and whenever the DNA polymerase needs bases to base pair, it can just suck in the materials because they are floating around. But again, the idea here is, where did they come from? They came from the food that you ate, because again, you are what you eat, right? So literally, if you eat chicken, those chicken materials will be inside your body, and you use those chicken materials. Technically speaking, is it really chicken, sir, when it's already in nucleotide form? Well, yeah, that is another argument for another time. But the point is that the materials of the food that you ate will also become your material, will become your new DNA and your new cells, and so on and so forth. So again, I just wanted to point that out because, um, again, I feel like when we discuss these things, we just you know get to the point of the DNA replication, and we don't really care to understand where these nucleotides are. And I feel like it's important that we really get the connection that, okay, that's how DNA replication is kind of connected to the concept of digestion or digestive system, which we have discussed in grade 8, and circulatory system, which we discussed in grade 9. See? So everything is connected. So it's better to study science this way. But anyways, once the DNA polymerase, so let's proceed. The DNA polymerase, let's just assume that it it would just go from this end, from the starting point, all the way up to the end of the strand of the DNA. Remember, the DNA is very long. And we call this part, this upper part, we call this the leading strand. Okay? So let me just mark this a little bit for you. So this strand here that's being built in the upper portion is called the leading strand. Okay? While this strand here, as you can see in the video, it's marked the lagging strand. Okay? It's the leading strand and the lagging strand. Why are they named like that? Well, you will see. So if we play this in the lagging strand, it's kind of a different game. Okay, it's kind of the different game. So remember, the leading strand is 5 prime to 3 prime, right? So in the lagging strand, so let's go backwards. So in the leading strand, if you go from the right part of your screen to the left, because remember this DNA polymerase is moving from the right end to the left end, it's 5 prime to 3 prime strand, right? So therefore, it stands to assume that when you go to the lagging strand, as you go from the right to the left, so right to left, it's 3 prime to 5 prime. Okay? This is where this concept is important in the narrative, because the DNA polymerase unfortunately can only do 5 prime to 3 prime. It cannot do 3 prime to 5 prime. So let's see how the DNA polymerase solves that problem. So again, 3 prime to 5 prime. The problem is, if that is 3 prime to 5 prime, guess what? Your DNA polymerase, if it starts here and goes that way, that direction is 3 prime to 5 prime. And again, what did we say? It cannot do 3 prime to 5 prime. Okay? It cannot do 3 prime to 5 prime. So, how would it solve that obstacle? Well, see, the primase moves ahead forward. Okay? And then the DNA polymerase also moves ahead forward. And then this is the clever bit. It builds the DNA polymerase, that segment, builds a DNA segment, that Okazaki fragment, backwards. Right? It builds it backwards. Why? Because again, if the DNA polymerase goes from the right end of your screen to your left, it will be moving 3 prime from the 3 prime end to the 5 prime end. And again, it cannot do 3 prime to 5 prime. So what it does is it instead moves ahead forward and then backtracks. Because if you backtrack, think about it, if forward is 3 prime to 5 prime, then backwards is 5 prime to 3 prime. So it now circumvents its limitations because it cannot do 3 prime to 5 prime. If I cannot do forward, then I will do it in backward style. And because of this, if you notice, the lagging strand is different because it does it. I'm sorry. The lagging strand is different because it does it in segments. So you see, it starts here and then creates this segment. Okay? So if you go back further, just to see the lagging strand, this segment here is what we call an Okazaki fragment. See, Okazaki fragment. So it does it in fragments. So what it does, what the cell does is it kind of tells the primase to move ahead forward and then attaches, and then the DNA polymerase now knows where to start. And then again, if it goes forward towards the left, it goes 5 prime to 3 prime. So it cannot do that. So it has to do backwards. And it builds one fragment. And after that, it now builds a different fragment. Right? So it's created in Okazaki fragments. Right? So take note how different that is from the leading strand. In the leading strand, all the DNA polymerase needs to do is start where it starts, right? And then after that, it just simply waits for, sucks in all the nucleotides that are floating, and then it will just simply attach them from 5 prime to 3 prime from the start all the way up to the end of the DNA in one continuous process, right? But in the lagging strand, it can't do that, right? So what it does is it instead builds it in Okazaki fragments. And again, it's important that you have a narrative here. Instead of saying, okay, it has the lagging strand has fragments, the leading strand has one continuous strand. Again, that's boring. What you need to do is you understand why the DNA polymerase has to do that. So think of it as a story. And the story is the DNA polymerase has a problem in the lagging strand because if it moves forward, just like in the leading strand, it will be going against what it can do. And what it can do is only 5 prime to 3 prime. It cannot do 3 prime to 5 prime. So to circumvent the problem, it makes it in fragments. Okay? So as you can see, in the you can imagine that in the leading strand, if this is the leading strand, right? If this is the leading strand, there's the primers here, okay, um, attached by the primase, and then the DNA polymerase simply builds all the way until it reaches the ends. So in the leading strand, there's only one primer, but in the lagging strand, because it has to do in fragments, again, it the primase has to attach a primer here, and then the DNA polymerase has to build, but again, backwards. That segment, that's an Okazaki fragment. After that, another primase would build another primer here, primer bases here, and then the DNA polymerase now builds it backwards. So it's in fragments, and then repeat. So it's like it moves forwards, backtracks, moves forward, backtracks, moves forward, backtracks, moves forward, backtracks. So therefore, in the lagging strand, there are many primers, primer bases that are inserted by the primase. But in the leading strand, there's only one primer replication. So this is where the next enzyme now takes into action. So because of the many primers that are in there, there's another enzyme whose purpose is just to remove the primers. Because if you think about it, the primers are RNA bases, and that's not good. So after the primers have been removed in all these sites, the DNA polymerase goes back to it and then base pairs it with DNA molecules this time. Because again, remember, the primers are RNA bases. So after attaching the primers, the nucleus has to remove those RNA bases because after all, these are RNA bases, not DNA. These are just temporary markers, so to speak. So the exonucleus removes them, and then the DNA polymerase goes back to where those primers, to the space where the primers were before, and then attaches the proper DNA bases, right? And then after that, the last step is this, which is you have the enzyme called the ligase. And the ligase, think of the ligase as uh, it it seals back the DNA, right? It seals back the DNA because when they base pair, think of it as the bonds are not that, shall we, for the lack of a better term, are not curated yet. So the DNA curates them, seals them more permanently. So the DNA ligase does that, and then now you have two new DNA strands. And the thing to note here is again, in that DNA strand, it's not one is not completely old and one is not completely new. One strand is old and the other strand is new. So it's a conservative process. Okay, so those are the things. So if we look at it in terms of processes, so there are different processes. First process is the DNA split. Second process is, so the helicase is involved. Second process is the DNA polymerase has to know where to start. So that's the goal. That's the purpose of the primase. It attaches RNA bases that constitutes what we call the primer segment. It's a primer. and then the DNA polymerase goes to it and then does its work. But again, in the leading strand, because it has to go from 5' to 3', it just starts from where it starts and then goes all the way up to the end of the DNA. But in the lagging strand, because if it starts from where the other end is and goes forward, it has to do 3' to 5'. And the DNA polymerase cannot do 3' to 5'. So it cleverly avoids that problem by going forward and then backtracking, because if forward is 3' to 5', then backward is 5' to 3'. So it goes forward and then backtracks, forward and backtracks. So building the DNA in segments in what we call Okazaki fragments. Now, let me address some nuances here. And then the nuance is like this, sir. Okay, the DNA therefore has two strands, right? And again, let me make this two like that. This strand is 3 prime. This strand is 5 prime. So therefore, the other strand is opposite. It's 3 prime to 5 prime. When this splits open, this is 5 prime and 3 prime, and the other end is obviously the same as the what is in the upper part, 5 prime to 3 prime. So again, take note, this strand has 5 prime to 3 prime. So therefore, when you build the second half, the missing half, let me call this, let's put some dot 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 just to know that this half is still missing and you are building out this half. If this is 3 prime and that's 5 prime, therefore what you will build is 5 prime to 3 prime. Get? Because in the original, 5 prime in the upper part, 5 prime to 3 prime in the upper part, 3 prime to 5 prime in the lower part. So therefore, when it splits, the upper part, what you will build is the 3 prime to 5 prime, if you go from left to right. But again, if you go from right to left, as in the video, then you start from the 5 prime to the 3 prime. So if this is the case, and you start here, and this is the start, so what happens is when the DNA polymerase gets into here, then it has no problem. It starts from the 5 prime, builds all the way to the 3 prime in one continuous go. So this is now the leading strand. What is being built is the leading strand. Again, if you go to lagging strand, because this was the bottom part, which is 3 prime to 5 prime if you go from left to right, 3 prime to 5 prime. So what you will build is 5 prime to 3 prime if you go from left to right. But in the video, it starts from here. So if it starts here, if it starts from the right side, so right to left is 3 prime to 5 prime. So it cannot do that. It cannot start here and go this way. So again, what it does, it moves forward here and then builds this in segments backwards. Because again, if this is 3 prime to 5 prime from right to left, then if you go from left to right, which is backwards, this will be 5 prime to 3 prime. Right? And then once it does that, it moves to the next section, builds that fragment backwards, and then moves that section, builds that fragment backwards, moves that section, builds that fragment backwards. Okay? That's why it has fragments. But, sir, if you are observant, you would have this question. You would say, sir, why not just wait until the DNA is completely split into two, like this? So I know the DNA is long, but imagine this is the whole DNA. If it's split completely into two, then here you will build the 5 prime to 3 prime, right? Here you will build 5 prime in this end, and then 3 prime in this end. So, sir, why not DNA polymerase will start here at the 5 prime, builds this in one segment, and then why not the DNA polymerase on the second strand start here? So if it starts here, it circumvents the problem. It's now starting at 5 prime to 3 prime and then go in one continuous direction. To be honest, yes, that is more efficient in terms of the solution, right? But you have to understand that this DNA is so long. So imagine if it's kilometers, and in fact, the DNA can actually circumvent the circumference of the Earth if we put all the DNA in your body. But the point is, it's so long that if you wait for the time that the whole DNA splits completely and then start at the other end, then the DNA strand would be entangled already. And you don't want that because the DNA might be damaged. So it's not wise. So conceptually speaking, this is more efficient, right? Just split the DNA into two, wait until the whole thing gets split, and then one DNA polymerase starts from one end, and then the other DNA polymerase starts from the opposite end. In that way, this DNA goes from 5 prime to 3 prime. This polymerase here goes from 5 prime to 3 prime, and the other polymerase goes from 5 prime to 3 prime, and then they just go from where they start all the way to where they end, and they don't have to build it in fragments. Again, conceptually speaking, that is more efficient, but again, you have to consider that the DNA is so long that if you wait until the whole DNA is unzipped completely, by the time you go to the other end, the other end is already split also, you would have a mess, right? So again, in that sense, the Okazaki fragments is a better way to go. Okay, so I just wanted to point that out. Then the next nuance, let's say, that I would want to point out is this idea that, again, let's go back to it. The DNA polymerase, again, can only do 5 prime to 3 prime, and it cannot do 3 prime to 5 prime. Okay? So why is this? Well, let's just have an analogy. And the analogy is that imagine that the DNA polymerase has a front and a back, and what it does is it can only move front first. So it can only move this way. So when it attaches to the DNA polymerase, it can only move that way. And then if you notice, the DNA nucleotides that the DNA polymerase has to attach has a shape, right? So I mean, it has a phosphate group, right? And then it's connected to a sugar, which is a deoxyribose, and then a nucleo. So this is the ribose sugar, or maybe just S for sugar phosphate group, sugar phosphate group, and then this is the nitrogenous base. Think of it as when this DNA polymerase sucks in, just like what you can see here, with that DNA polymerase sucks in those nucleotides inside, right, from the surrounding material, because again, these DNA polymers are just these DNA nucleotides are just floating in the material. Think of it as the gate which in which this enters to the polymerase has a particular shape. Think of it as it has a particular shape, and that shape is like this, such that this has to fit in the opening before it gets attached, right? So the problem, so now you can see the what the problem is. The problem is if you so remember this is the DNA polymerase, and if you remember our counting, this is carbon number 5 here, and this is carbon number 3. So go back and watch the video when I discuss the 3 prime and 5 prime if you do not understand what I'm talking about here. But um, this is carbon 5 and this is carbon 3. So therefore, in this direction, in this idea, this is the 5 prime end and this is the 3 prime end. I'm sorry, I actually the DNA polymerase has to be attached like that, right? So let me uh, rearrange this. Let me rearrange this. The opening is something like this. I hope I get this right. Like that. So it has a particular shape. So um, if you notice, how will this fit in there? How will this fit in there? It will only fit in this shape if this rotates like that and enters this case, right? If it rotates like that, it enters that. So what happens if it rotates like that? So therefore, this end here will be 5 prime, and this end will be 3 prime. So I will rotate this and see if I have to fit this in here. I'm sorry. Well, yeah. If I have to fit this in here, I have to rotate it like that. And therefore, this edge is 5 prime. See, if I rotate this like, maybe I'll have to get another, uh, this one. So if this is the phosphate group, and then you have the sugar group, and then you have the nitrogen space. And again, this end here is the 5 prime end and the 3 prime end. If I have to rotate this like that so that it fits in here, you see this end is 5 prime, that end is 3 prime. And since the polymerase can only go forward, so when this enters and attaches, this is 5 prime to 3 prime. Then when it moves and then another attaches, it still goes 5 prime to 3 prime. So this polymerase can only attach can only build the bases from 5 prime to 3 prime. Okay? Because if I will, for example, rotate this to force that it will be 3 prime to 5 prime, see, it will not fit in the slot. It will not fit in the slot. Okay? So maybe let me. I realize that it's not according to what the video shows you. So let me repeat that again. Um, if this is the DNA, let me turn this sideways first. If this is the DNA, so you have the phosphate group, and then you have the sugar group, and then you have the nitrogenous base. And again, this is the 5 prime carbon, and this is the 3 prime carbon. And if you do the opposite direction, which is like this, phosphate, sugar, and then nitrogenous base, and this is the 5 prime end, and this is the 3 prime end. See, if this is the DNA polymerase, and again, I realized that I did not draw the DNA polymerase according to how it should look like in the video. So I'm redrawing it right now. This is the DNA polymerase, this is the front, and this is the back. And then the DNA polymerase has to move forward. It has to move like this. Again, the idea is when it sucks in the nucleotides, it has a door that allows it to the DNA to come in, and then that door has a certain shape, and fortune and the shape is like this. Again, I'm like this. So you see when I'm sorry, I did it again. Sorry, I did it again. But this time I will do it correctly. So you have here the um, phosphate group. Sorry about this. Let me, because it's difficult for me to draw. So you have the phosphate group, then you have the sugar, then you have the nitrogenous base. And again, this is the 5 prime end, this is the 3 prime end. And again, if we were to force the issue, then I would have to draw something where the phosphate is here, then the sugar is here, and then the nitrogenous base is here. And again, this is the 5 prime, and then this is the 3 prime carbon. right? So which means that this has a particular shape, and that particular shape is like this. So think of it as the square part is in this part, and then the circle part is in this part. So this will fit, see, inside and can enter the polymerase. But this cannot, because this, think of it as this square side is not doesn't fit in the circle side, and then this circle side doesn't fit in there. This can fit, but it has to rotate. It has to not rotate. It has to do like this. If this is that, it has to do like this. Flip, right? And then it will now fit here. So therefore, this can fit. So if this can fit in here, notice that this side is 5 prime, that side is 3 prime. I mean the 5 carbon and the 3 carbon. So as a result, this is 5 prime to 3 prime. And if the polymerase moves forward, so it's 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime, it cannot go the opposite direction. Because to attach a DNA polymerase in the opposite direction, it means that you have to have the 3 prime to 5 prime end on the opposite side. So if you want to force the issue, if you want to force the issue ng 3 prime to 5 prime, so dapat you mirror this like that. So if you mirror this like that, it will be like this. Phosphate. Right? So because in this case, remember this is the 5 prime end here. This is the 3 prime end. So if you want this, if you want 3 prime to 5 prime, you have to fit this. And again, it doesn't fit because for example, the circle side is on the right side, but this one is on the left side. The square side is on the right side, but here a rectangle side is in the left side. So it will not fit. So again, this will not fit, but this will fit. So 5 prime to 3 prime fits, 3 prime to 5 prime doesn't fit. And since the DNA polymerase can only move forward, it cannot move backwards, it has no choice. So 5 prime to 3 prime doesn't fit, only 3 prime to 5 prime. Okay? So again, when we evolve to be like that. We evolve that our DNA polymerase can only do 3 prime to 5 prime. Now, could it not have been the other another way? Meaning, could it not have another way? Meaning, could it not have evolved such that the DNA polymerase can do two directions, can do 3 prime to 5 prime and 5 prime to 3 prime? Yes, it could have evolved like that, but we did not evolve like that. So again, you have to remember that evolution is kind of small accidental changes. It's just so happened that we have this machinery that called DNA polymerase, and the DNA polymerase unfortunately cannot do 3 prime to 5 prime. So it has to circumvent the problem, right? And if you think about it from the evolutionary perspective, if I'm a cell and I have to evolve DNA polymerase that can go 5 prime to 3 prime, and then another DNA polymerase that can go 3 prime to 5 prime, that means I have to go another million or thousands of or hundreds of millions of evolution to develop that other kind of DNA polymerase that can build 3 prime to 5 prime. So, and again, that's in if in terms of it's resource intensive. Instead of just, you know, why don't I just use the same machinery, DNA polymerase that can only do 5 prime to 3 prime, but I'll just do a clever way of doing the other strand, the lagging strand, which is in terms of Okazaki fragments. So apparently that works for the cells. But could we have evolved it? Could we have evolved differently? Yes, we could have evolved into a configuration wherein we have two types of DNA polymerase. One DNA polymerase can do 5 prime to 3 prime, and the other DNA polymerase can do 3 prime to 5 prime. But as it stands now, our DNA polymerase, in fact, even our RNA polymerase, which we will be talking about in the protein synthesis, can also only do 5 prime to 3 prime. Okay? So these are the nuances for this video. Now let's go to the other video, so the realistic one. So basically, this is just the same process. It's just that this one is um, what really happens in reality, right? So in the real world. So, but first, let's since it's already here, let's just discuss some of the some of prerequisite concepts. Like this is a DNA. So if you can see, it's not squares and lines anymore, because these are realistic kind of molecule atom by atom representation. So if you can see, that's the DNA, and the DNA attaches to these certain proteins called histones. And if you notice, the DNA is being coiled around, and even the coils are coiled. So it's the DNA is coiled into smaller coils. The smaller coils is attached into bigger coils, and the bigger coils even form even bigger coils. And that is now what you call the chromosome, right? So if we when we zoom this out, we can see that these actually make up your chromosomes, right? So these are coils upon coils upon coils. Think about it as it has different structures. So one DNA strand is wrapped around histones. The wrapped histones and DNA assembly also gets wrapped into coils, and then it becomes the chromosome. And again, in terms of the narrative, you have to think of thread, right? If you have a long piece of thread, how do you organize thread? Do you just have the thread, put it in the drawer? Of course not. It will entangle itself. So if you have long structures like a thread or a piece of rope, you don't store it lying around. You organize it some way. In the case of rope, you kind of form circles, it loops in it. In the case of thread, you loop it around something, right? So that it holds the the thread strands without entangling. It's the same thing for the DNA, because the DNA is a long piece of strand. So you have to do this, and that's what you call the chromosomes that you see. And again, there are 23 of them. In fact, 23 pairs also. So that's your DNA, and then this is the division. But we are into the DNA replication part. So this is really what happens. So if you look at that, sure, it's kind of very different from what we talked about, because in the simplified video, we have steps. Step 1, split open the DNA into a replication fork. Step 2, primase attaches primers to certain sites. Step 3, DNA goes to the where the primers are and then start the DNA replication. In the leading strand, it's one continuous strand from start to end. In the lagging strand, it's moving forward, backtracking into one fragment, and then moving forward again, backtracking into another fragment, so on and so forth. Then after that, the exonuclease goes into and removes the primers. And then after that, the DNA polymerase goes back to the spaces left by the primers and then attaches DNA polymerase. And then after that, the ligase goes into action and seals the whole thing. It's much more organized in our head. Obviously, you have to split it open. You have to tell where to start. You have to do the base pairing, and then you have to remove the primers. You have to replace the primers with proper DNA nucleotides, and then you have to seal the whole thing, right? And again, it helps us organize our thoughts. But the reality is, it's not like that. It's not step 1, step 2, step 3. It's happening at the same time. So if you notice here, what is happening, sir? This strand here. I'm sorry, I couldn't see my tool. Okay, here. So if I will show you my video, the video feed, if you see this here, this is the helicase. This is the DNA polymerase. At the end, there's the ligase. This is a DNA polymerase. At the end, there's a bit that's called the ligase, and it's happening all at the same time. What do you mean it's happening all at the same time? If I play, sorry, I need to choose cursor. If I play, while the helicase is, splitting the two strands, the DNA polymers are working. The exonucleus are also working. The ligase are also working. The primates are also working. So it really happens very, very fast. And there's no step one, there's no step two. Okay? So let me point out some subtleties here. So number one, when you do DNA replication, again, you have to have the lagging strand and the leading strand. And that leading strand, again, it just starts from where it starts and then goes all the way up to the end. In the animation, the leading strand was the upper strand. In here, the leading strand is the lower strand. So if you look at this part here, this is the leading strand here. The leading strand is being built here. So if you notice, the DNA polymerase, it has no movement. It just starts where it is, right? And then builds, 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 builds. How do I know that it's building, sir? Well, I can pause the video and notice that, let me remove some of the markings so that it's not confusing. Notice that this here is the DNA. And then when it goes back, it's split into two strands. This is the one strand number one, and this is strand number two. Okay, again, it's much more complicated to see because it's more realistic, but this is one single strand, and then the helicase splits it into two, this strand and that strand. And then if you notice this, the DNA polymerase builds the other strand such that when it comes out, notice when it comes out, it's already one whole DNA, one new DNA already. Okay? And if you notice, this DNA polymerase doesn't really have any movement. It just stays there. Why does it stay there? Well, it just stays there because if this is the DNA strand, it just starts here, builds, 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 builds as the DNA moves past it until it reaches the end. It doesn't have to have any movement, right? But if you look at the other strand, let me backtrack a little bit. If you look at the other strand here, a lot of things are going on. See? Pack, it ends and then builds another fragment. See? That's one Okazaki fragment. Once it's done, it builds another fragment. That's another Okazaki fragment. So on. So see, once it starts, it builds another fragment here. So it's much better to see in this case. So let us move a little bit. See, pause. So you see, this case, this one is one fragment. So it starts here, goes back. And this here is one Okazaki fragment, meaning this is the lagging strand. And then once that is done, another fragment will be built, this fragment here. Okay? So if we remove the markings, maybe you can see it, or maybe let me put that back in. So again, this is one fragment, that's one Okazaki fragment, and it's nearly done. That's one DNA polymerase. See, it starts here and then goes back. It's nearly done. Once it's done, it will have another process that builds this fragment here. This is think of it as Okazaki fragment number one, Okazaki fragment number two. So if I play this, see what happens. Play this video again. Go. So now another Okazaki fragment is being built. See? That's another fragment again. Then once that is done, another fragment is built again. But if you notice, it's very busy. It's not like step one has to be done first. The splitting must be done first before the putting of the primates, before the DNA polymerase does its job, before the exonuclease removes the primers, before the DNA polymerase attaches DNA nucleotides to where the primers used to be. It's after that the ligase goes into work. It happens much more at the same time. So you can imagine here, look at it. The DNA again is one whole strand here. It's being split into two. strand number one, strand number one, strand number two. The strand number one is the leading strand. Then you have the DNA polymerase complex here. And then when it comes out, notice when it comes out, it's already one whole new DNA. What does that mean? It means that when this comes out here, the ligase has already sealed this DNA. So therefore, you can imagine, although I can't see it, that somewhere here must be the ligase enzyme. And it must have been after the DNA polymerase now uh, base pairs, and then the um, after that, because remember here there's only one primer end, not a lot of primers to remove, so there's no need for the exonuclease here, only at the start. After that, the ligase would seal this whole thing, so when it comes out, it's one whole new DNA. See, so again, um, this is why I had a hard time to show. I had a hard time deciding which video to show. Is it the realistic one or the more simplified one? The simplified one, you can it's easier to understand, but it's not it's simplified that it loses some of the ideas. But this is really what is happening. See, the fragments are being built um, like that. Now, if I was the animator, I would have done one more thing, which is to show the nucleotides that are floating being sucked in by the polymerases. But apparently here it's not done. But the idea is like that. See, do you see the green one? Do you see the green one? See this green one floating? And then it attaches here. Or not the green one, but um, the blue-green one. So you notice this one, this here? It attaches like that. Before it was not there. So 1, 2, 3, go. There, it attaches. I suspect that's a primase because this is about to start a new Okazaki fragment. See, it has, because this Okazaki fragment here is about to end, so it has to start another Okazaki fragment. And notice what it does. It attaches here. So I suspect that that's because it's attaching the primer bases here, the RNA primer bases, so that in the next step, the DNA polymerase can start this fragment anew. Notice when this second fragment is started, this second Okazaki fragment is started, the first Okazaki fragment is nearly done. So it's not like this first fragment must be done first, and then the same DNA polymerase will jump to the next fragment. There are two DNA polymerases working at the same time, right? So that one DNA polymerase is nearly done in making the first fragment. The second DNA polymerase is starting already the second fragment, the second Okazaki fragment. So again, as I said, as I told you, there are more nuances in the. So it's actually three DNA polymerases. So if you can really see, this is DNA polymerase 1, and it's just steady because it's in the leading strand. It doesn't have to do anything. But then you have DNA polymerase second and third, and if you notice the second and third, they alternate. So when this builds the first fragment, Okazaki fragment, the second fragment, the second DNA polymerase is preparing the next Okazaki fragment. Once it's done, they switch, and then the second, this DNA polymerase will now prepare the third fragment. So let me play that once back, and I want you to notice that these two switch as time passes by. So see, this is the fragment. This is the polymerase doing the fragment, and then now see it switches. See, now it's now this one. So that's how complicated the process are. But again, science is like a simplification of things. So it's nice to know that this is how it is really done. But in terms of organizing our thoughts, it's better to think of, one thing is splitting the DNA, the helicase, then the primase is building where to start, building the primers to tell the DNA polymerase where to start. The DNA polymerase goes in there and does its job. Again, in the leading strand, it just goes from start to finish in one continuous strand. In the lagging strand, it has to do a specific maneuver, which is move forward and then do the whole thing backwards. Because again, it has a limitation, and that limitation is that it cannot do it cannot do 3' to 5' direction. It's 5' to 3'. It can only do 5' to 3'. And I'm emphasizing that because later on in the DNA in the protein synthesis, it's also the same thing with the RNA polymerase. It can only go 5' to 3'. And then after that, the bases, the primers must be removed through the exonuclease. And then after the after the primers are removed, the DNA polymerase goes back to the spaces left by the primers and then builds in the DNA bases. And then the ligase seals the whole thing. It's much more organized in our head, but in reality, it's a little bit more chaotic because most of those things are happening at the same time. But in terms of organizing it into steps so that it's more digestible in our heads, it is done like that. Okay, so that is it for the video about DNA replication.